Hello students, welcome to EPG Pathshala. I am Dr. Ravi Toteja, Associate Professor in Zoology from Acharya Narendra Dev College, University of Delhi. Today we are going to discuss about a module, Overview of Immune Response under paper Immunology. The learning objectives are how the branch of immunology has originated, basic concepts of immune response, what is specific and non-specific immunity, understanding of active and passive immune response. To begin with, we will start with the historical perspective of immunology. The word immunity means to free ourselves from the microorganisms and is used in the context of being free of the burden of taxes or military conspirations. The history of immunology is really slightly more than 100 years. If one considers Louis Pasteur as the father of immunology. From early writings, it is clear that primitive man knew about the disease and its consequences. The records of the presence of pestilence and the disease can be found in Babylonian epic of Gilgamesh. In other more recent writings from old dynasties of ancient Egypt, one finds even more description of the disease. From these writings, it is apparent that man knew that once he had survived the disease, he will normally not able to get that disease again. The science of immunology grew from this knowledge that those who survived the infection rarely contracted that disease again. This was an observation that was made long before the establishment of germ theory of disease. In 430 BC, Thucydides recorded that while the plague was raging in Athens, the care of sick was being taken by the individuals who had already contracted the disease and recovered. Beginning around 1000 AD, the ancient Chinese practiced a form of immunization in which they inhaled dried powders derived from the crust of smallpox lesions. Around the 15th century, a practice of applying powdered smallpox crusts and inserting them into the skin became common practice. This process is referred as variolation and became quite common in the Middle East. Lady Mary was the wife of the British ambassador to Turkey. She was a smallpox victim. She variolated both her sons and daughter in 1717 and 1721 respectively. Initially there was much resistance and criticism by the higher society in England, but later on the permission to vaccinate the children of Prince and Princess of Wales in 1772 dramatically promoted the adaptation of this method in England and in other parts of Europe. Ultimately, the process was popularized in Great Britain. It was strongly opposed by the church and was highly downcasted, particularly if one were Christian. By the second half of the 18th century, it was noticed that women who milked cows were safe from smallpox disease. Numerous undocumented accounts show that connection was made between contact with cowpox virus and protection from smallpox. Taking clue from this, Edward Jenner started thinking about vaccine. After years of experimenting, Edward Jenner carried out a famous experiment. He deliberately exposed the boy who had recovered from the chickenpox to virulent variola virus 2 months later and found that child was protected, showing only a mild inflammation around the site where the virus was injected. Jenner wrote a paper explaining his experiments and wanted to report his first case study in the Royal Society of London. His study was rejected. He then went to London to demonstrate his theory. No one would submit to his vaccination. Discouraged, Jenner returned to Berkeley and he published The Origin of Vaccine by Inoculation, describing how cowpox virus was prepared and used to protect healthy persons against smallpox. Vaccination, the word Jenner invented for his treatment, was adopted by Louis Pasteur for immunization against any disease. Louis Pasteur developed germ theory of disease. Pasteur work led him to the development of pasteurization as means to stop fermentation. He was the first one to grow in vitro the culture of infective organism of fall cholera. In his experiment, he inoculated the culture of bacteria to chicken. Some of them survived and many of them died due to infection. He then went for the vacation. On coming back, he injected the leftover culture to the chickens who had survived from the previous infection. To his surprise, no one got infected. He reasoned that it could be because of, first, the culture which he had used for the second time has lost its virulence because of aging. There is development of resistance to infection in the chicken because they had survived the first infection. This was the start of using attenuated microbes for providing immunity against infection. He also found that rabies was transmitted by agents so small they could not be seen under a microscope, thus revealing the world of viruses. As a result, he developed technique to vaccinate dogs against rabies and to treat humans bitten by the rabid dogs. Robert Koch, 1843 to 1910, a German physician who worked on anthrax and found out the causative agent of an infectious disease. In 1882, Koch was able to demonstrate that the germ theory of disease applied to human ailments as well as animals. When he isolated the microbes that caused tuberculosis, Koch's postulates are still used to identify infective organisms. He was awarded 1905 Nobel Prize. Von Behring. Emil A.V. Behring, a student of Koch, along with Kitasato and Wernicke, discovered antitoxin for diphtheria and tetanus and applied it as therapy. Emil von Behring and Kitasato were able to demonstrate passive immunity when they took serum from animals infected with diphtheria and injected them into healthy animals. These same animals were found to be resistant to the disease. Ultimately, these serum factors were acknowledged in 1930 as antibodies. In 1901, von Behring was awarded first Nobel Prize in Physiology. Paul Ehrlich. Ehrlich hypothesized that antibodies are specialized molecular structures with specific receptor sites that fit each pathogen like a lock and key. Thus, the first insight that the body has a specific defense system was introduced. He also realized that this powerful effector mechanism, which is used in host defense, would cause severe tissue damage if turned against the host, and he termed this as autotoxicus. He shared 1908 Nobel Prize with Mechnikov. Mechnikov was an embryologist and studied the starfish development. He was the first one to found phagocytosis and birth of cellular immunology. Phagocytosis is the process of eating any foreign body. He shared Nobel Prize with Ehrlich in 1908. Then comes Brunet. He described the clonal selection theory to explain tolerance, and he received Nobel Prize for the discovery of acquired immunological tolerance. Rejection of donor grafts was due to an immunological reaction. The classification of the immune response and the science of immunology did not progress in a systematic or chronological order. Nevertheless, one scientist had a basic understanding of the cellular and humoral branches of the immune system. What remained was the identification of the various components of this intrinsic system and the mechanism of their interactions. This could not have been accomplished without the concomitant development of molecular biology and genetics. Selective theory. Selective theory states that an antigen reacts with an already existing antibody. The earliest conception of the selective theory dates back to the Paul Ehrlich. In order to explain the origin of serum antibody, he proposed that cells in the blood express a variety of receptors, side chain receptors, that could react with the infectious agents and inactivate them. Ehrlich proposed that binding of a receptor to an infectious agent was like a fit between a lock and key, and he suggested that interaction between the infectious agents and the cell-bound receptor would induce the cell to produce and release more receptors with the same specificity. The specificity of the receptor was determined before its exposure to the antigen, and the antigen selected the appropriate receptor. All aspects of the Ehrlich theory would be proven correct with the minor exception that the receptors exist as a both soluble antibody molecule and the cell-bound receptor. Next is instructional theory. Instructional theory suggests that antigen play a pivotal role in determining antibody specificity. According to this theory, a particular antigen would serve as a template around which the antibody would fold. The antibody molecule would thereby assume the conformation complementary to the antigen. The most accepted theory is the clonal selection theory. Clonal selection theory is the most accepted theory given by Journey, Talmadge, and Brunet. According to this theory, an individual lymphocyte, BOT, expresses membrane receptors that are specific for different antigen. This specificity is determined before the lymphocyte is exposed to the antigen. Binding of an antigen to its specific receptor activates that particular cell, leading to its proliferation into a clone of cell that express the same immunological specificity as the parent cell. Now coming to the types of immunities. There are two types of immunities, innate and adaptive immunity. The immune system is a system of biological structure and processes within an organism that protects organisms against disease. It detects a wide variety of disease-causing agents, from viruses to parasitic worms, and needs to differentiate them from organisms' own healthy cells and tissues in order to function appropriately. Simple unicellular organisms such as bacteria have an enzyme system that protects against viral infections. Other basic immune mechanisms evolved in ancient eukaryotes and remain in their modern descendants such as plants and insects include antimicrobial peptides called defensins and phagocytosis and the complement system. This type of protective mechanism is present by birth and is known as innate immunity, and this is non-specific also. Jawed vertebrates, including humans, have complex defense mechanisms. The typical vertebrate immune system consists of many types of proteins, cells, organs, and tissues that interact in a complicated and vigorous network. As part of this more complex immune response, the human immune system adapts over time to recognize specific pathogens more competently. This adaptation process is referred to as adaptive immunity or acquired immunity and generates immunological memory. Immunological memory generated from a primary response to a specific pathogen provides an enriched response to secondary encounters with the same antigen or pathogen. This process of acquired immunity is the basis of vaccination. Primary response can take 2 days to 2 weeks to develop. After the body gains immunity towards a certain pathogen, when infection by that pathogen occurs again, the immune response is called the secondary immune response. Now coming into details of innate immunity. Microorganisms are pervasive. They are in the air we breathe and on the food we eat. Thus, our epithelial surfaces, skin, respiratory tract, gastrointestinal tract, urinogenital tract are continuously exposed to microorganisms. Disease occurs when microorganisms are able to invade epithelial surfaces. In view of our constant exposure to microorganisms, it is surprising that normal, healthy person experiences long infection-free periods and that infections are the exceptions rather than the rule. This resistance of epithelial surfaces to invasion is a characteristic which is present from birth and is therefore called innate immunity. The elements of innate immune system includes anatomical barriers, secretory molecules, and cellular components. Among the mechanical anatomical barriers are the skin and the internal epithelial layers, the movement of intestines, and the oscillation of bronchopulmonary cilia. Associated with these protective services are chemical and biological agents. Anatomical barriers to infection. First one is the mechanical. The epithelial surfaces form the physical barriers which are impermeable to most infectious agents. The skin acts as our first line of defense against any invading organisms. The desquamation of the epithelial is also helps in removing bacteria and other infectious agents that have attached to epithelial surfaces. Movement of cilia or peristalsis helps in keeping air passages and gastrointestinal tracts free from the microorganism. The flushing action of tears and saliva helps in preventing infections of the eyes and the oral cavity. Second, chemical. Fatty acids in sweat inhibit the growth of bacteria. Lysosome and phospholipase found in tears, saliva, and nasal secretions can break down the cell wall of bacteria. The acidic pH of sweat and gastric secretions prevents growth of bacteria and many other microorganisms. Defensins, which are low molecular weight proteins found in the lung and gastrointestinal tract, are known to have antimicrobial activity. Surfactant in the lung act as opsonin, molecules that promote phagocytosis. Third one, biological. The normal microbial flora of the skin and the gastrointestinal tract prevents the establishment of pathogenic bacteria by secreting toxic substances or by competing with the pathogenic bacteria for nutrients or attachment of the cell surfaces. Now coming to humoral barriers of infection. The anatomical barriers are very effective in preventing colonization of tissues by microorganisms. However, when there is a damage to tissue, the anatomical barriers are infringed and infection may occur. Once infectious agents have breached the tissue, another innate defense mechanism comes into play, and that is acute inflammation. These humoral factors are found in serum. First one is complement system. The complement system is a major humoral non-specific defense mechanism. It is responsible for the increased vascular permeability, recruitment of phagocytic cells, and lysis and opsonization of bacteria or microorganisms. Next is coagulation system. Depending on the gravity of the tissue injury, the coagulation system may or may not be activated. Some products of the coagulation system can contribute to the non-specific defense, because of their ability to increase vascular permeability and act as chemotactic agents for phagocytic cells. In addition, some of the products of the coagulation system are directly antimicrobial. Lactoferrin and transferrin. These proteins, by binding with an iron, which is an essential nutrient for bacteria, downregulates the growth of bacteria. Interferons. Interferons are proteins that can control virus replication in cells. Lysozyme. Lysozyme breaks the cell wall of bacteria. Interleukin, and especially interleukin 1. IL-1 induces the production of acute phase proteins which are antimicrobial and they can opsonize bacteria. Now coming to cellular barriers to infection. Part of inflammatory response is the employment of polymorphonuclear eosinophils, neutrophils, macrophages to the sites of infection. These cells are the main line of defense in non-specific immune system. Neutrophils. These cells are engaged to the site of infection where they phagocytose invading microorganisms and kill them. In addition, they also contribute to collateral tissue damage that occurs during inflammation. Macrophages. Tissue macrophages function in phagocytosis and intracellular killing of microorganisms. Furthermore, macrophages contribute to tissue repair, act as an antigen-presenting cells, APCs, which are required for the induction of specific immune response. Natural killer cells, NK cells. NK cells can non-specifically kill virus-infected and tumor cells. These cells are important in non-specific immunity to viral infections and tumor surveillance. Eosinophils. The granules of eosinophils have lytic properties that helps in killing of certain parasites. Phagocytic barriers. Phagocytosis. It has derived from the Greek word phago, meaning eating, and cyte meaning vessels, and osis meaning process. So, phagocytosis is the cellular process of engulfing solid particles by the cell to form an internal phagosome. Phagocytosis is a specific form of endocytosis involving the vesicular internalization of solid particles such as bacteria or other microbes. Phagocytosis in the immune system is a major mechanism used to remove pathogens and cell debris. Bacteria, dead tissue cells, and small mineral particles are all examples of objects that can be eliminated by the process of phagocytosis. Inflammatory barriers. When the body is injured, a sequence of events is started that ultimately leads to the repair of the site. The inflammatory response is the body's natural response that occurs immediately following tissue damage. Its main function is to defend the body against harmful substances, dispose of dead tissue, and to promote the renewal of normal tissue. The inflammatory reaction is characterized by 4 distinct signs, each of which is due to a physiological response to tissue injury. First is dolor, which means pain, tumor, swelling or edema, rubor, that is redness, calor, or the heat. The inflammatory reaction is the combination of a number of overlapping reactions within the body. Although a lot of these occur simultaneously, but a certain order of events are: first one, tissue injury. Tissue damage may occur because of number of reasons. Then there is a release of chemicals. When the cells of a tissue are injured, there is a release of number of chemicals that initiate the inflammatory response. Examples of these are kinins, prostaglandins, and histamines. These chemicals work collectively and are responsible for increased vasodilation, that is widening of blood capillaries, and permeability of the capillaries. This leads to increased blood flow of the injured site. These substances also act as a chemical messenger that attract the body natural defense cells. This process is known as chemotaxis. Some chemicals also increase the sensitivity of the pain fibers in the area, and so the area becomes painful. Then there is a leukocyte migration. Chemotaxis leads to the migration of certain white blood cells, leukocytes, to the damaged area. Two types of leukocytes that are predominantly involved in the inflammatory response are macrophages and neutrophils. Neutrophils are first to migrate to the injured site and function by neutralizing harmful bacteria. Macrophages aid the healing process by engulfing bacteria and dead cells and ingesting them so that the area is clear for new cells to grow. They arrive at the injured site within 72 hours of the injury, and many remains in the area for weeks after the injury. Adaptive immunity, acquired immunity or specific immunity. The adaptive immunity is comprised of highly specialized immune cells and the processes that remove or check the pathogenic growth. This type of immunity works to protect and heal the body when the innate immunity fails. It provides the body with the ability to recognize and remember pathogens and/or antigens. This mechanism allows the immune system to mount stronger attacks each time the pathogen is encountered, thus preparing itself for future challenges and preventing reinfection by the same pathogen. The four hallmarks of the adaptive immunity are immunological recognition, self-non-self discrimination, immunological specificity, and immunological memory. An immune response must recognize a microorganism as foreign, non-self, as distinct from self, respond to a microorganism by production of specific antibodies and specific lymphocytes, mediate the elimination of microorganisms. An agent which evokes an immune response is called an immunogen. The term antigen is applied to a substance which reacts with antibody. Immunity can be of two types, active and passive. And there is third type also, which is called as delayed immunity. Active, passive, and delayed immunity are all variations on the operation of the immune system, whereby antibodies are produced in response to the presence of an antigen considered to be foreign. Active and passive immunity are versions of what is known as antibody-mediated immunity. That is, antibodies bind to the antigen, and this binding further stimulates the immune system to respond to the antigen threat. Antibody-mediated immunity is also called as humoral immunity. Active immunity. Active immunity ensures, due to the production of antibodies, antibodies are produced as a result of the presence of the target antigen, either as part of an whole infected organism or because of the introduction of specific antigen. The immunity is provided by an individual's own immune system. The type of immunity invoked by the active response tends to be permanent. The immune system has a capacity for memory of the antigen. If presented with the same antigen challenge again, the immune system responsible for the formation of the corresponding antibody is rapidly elicited. An example of active immunity is the injection into healthy individuals of the disabled toxins of bacteria such as Corynebacterium diphtheriae, the agent causing diphtheria, and Clostridium tetani, the agent that causes tetanus. Passive immunity. Passive immunity is also because of the presence of antibody, but the particular individual does not produce the antibody. Rather, the antibody is produced in someone else and is introduced to the recipient. An example is the transfer of antibodies from a mother to the unborn child in the womb. Such antibodies confer some immune protection to the neonate in the first 6 months following birth. Indeed, the transient nature of the protection is a hallmark of a passive immunity. Protection fades over the course of weeks or a few months following the introduction of the particular antibody. For example, a newborn carries protective maternal antibody to several diseases, including measles, mumps, and rubella. But by the end of the individual's first year of life, vaccination with the MMR vaccine is necessary to maintain the protection. Another example of passive immunization is the administration to humans of tetanus antitoxin that is produced in a horse in response to inactivated tetanus toxin. This procedure is typically done if someone has been exposed to a situation where the possibility of contracting tetanus exists. Rather than rely on individual's immune system to respond to the process of the toxin, neutralizing antibodies are administered right away. Delayed immunity. A third type of immunity, which is known as delayed immunity, is a different sort of immunity. Delayed immunity is also called cell-mediated immunity. Here, immune components called T cells bind to the surface of the cells that contain the antigen on their surface. This binding triggers a further response by the immune system to the foreign antigen. The response can involve components such as leukocytes. An example of delayed immunity is the tuberculin test or the Mantoux test, which tests for the presence of Mycobacterium tuberculosis, the bacteria that causes tuberculosis. A small amount of bacterial protein is injected into the skin. If the individual is infected with the bacteria or has ever been infected, the injection site becomes inflamed within 24 hours. This response is delayed in time relative to the immediate response of antibody-based immunity, hence the name delayed immunity. So students, let us summarize what we have learnt in this module. History of immunology dates back to Thucydides, where he wrote in 430 BC that only those who had survived the plague can nurse the sick because they would not contract the disease a second time. Chinese and Turks in 15th century started the process of varulation in order to protect themselves from diseases. Lady Mary Wortley Montagu virulated her children, and the technique was popularized in Great Britain. Vaccination, the word Jenner invented for his treatment, was adopted by Louis Pasteur for immunization against any disease. Louis Pasteur developed his germ theory of disease. Later, Mechnikov described the roles of cells, and at the same time, Behring described role of serum components in providing the immunity. Out of various theories of immune response, clonal selection theory of Brunet is widely accepted theory of generation of immune response. Vertebrates have two types of immunity: innate, which is non-specific, and adaptive, which is specific. Innate immune response begin much more quickly than adaptive ones, and therefore constitute the first line of defense after the host has been invaded by a pathogen. Innate immune response include various barriers such as anatomical, physiological, cellular, phagocytic, and inflammatory. Adaptive immune response exhibit four attributes: specificity, diversity, memory, and recognition of self and non-self antigen. Active, passive, and delayed immunity are all variation on the operation of immune system, whereby antibodies are produced in response to the presence of antigen considered to be a foreign. Active immunity occurs due to the production of an antibody as a result of the presence of target antigen, either as a part of an intact infective organism or because of the introduction of specific antigen in the form of vaccine. The immunity is provided by an individual's own immune system. Passive immunity is also because of the presence of antibody. However, the particular individual does not produce the antibody, rather the antibody which has been produced in someone else is introduced to the recipient. Thank you.